Good morning, everyone. Today we are going to discuss about microbial flora of milk and milk products. We know that various kinds of microorganisms enter the milk supply chain at various steps of production, processing, and distribution of milk or milk products. These microorganisms, they bring about undesirable changes in the milk or milk products. There are two impact of these microorganisms within the milk and milk products. Number one, spoilage of the product leading to loss of the shelf life of the product. Number two, if it is a pathogen, whether it is the organism itself or its toxin, such as enzymes and all, if they are present, they are having impact on the health of the consumers. So we need to, as a practice, when we talk about the clean milk production, we try to keep the milk and milk products free from these harmful microorganisms that are harmful in two ways. One, harmful to the product itself because there is loss of shelf life, and second is harmful to the human consumers, wherein there is a disease condition or upset in the normal health of a human consumer. Then, when it comes to the different kinds of microorganisms that are present in the milk and milk products, of all these microorganisms known, bacteria, virus, fungi, rickettsia, protozoa, chlamydia, the prions, when I talk about all these microorganisms, the most important and highly significant contributor is the bacterial contamination. That is why when we discuss about the microflora of the milk and milk products, predominantly it is bacteria, followed by fungi, and other very in a miscellaneous way these microorganisms develop. Say for example, viral infections. The viral contaminants could enter the milk supply chain. They may affect the human health due to the poorer surveillance and monitoring systems. Often we miss viral entities in the milk and milk product supply chain. Okay, when it comes to rickettsia, chlamydia, same thing is going to apply. Not much known about these microorganisms in the milk and milk products. Okay, even if they are there, their contribution compared to the bacterial flora is less. So whenever we talk about the microflora of the milk and milk products, it is predominantly of bacterial origin. So that's what is being discussed. Now, we know that milk is a highly nutritious medium. As it is nutritious to the mammals and other animals, young one especially of that particular species, it is also nutritious for the microorganisms. There is a required macromolecule composition, there is a required pH, and the temperature if it is provided, these organisms luxuriantly undertake the binary fission, leading to geometric increase in the number that would in due course of time affect the shelf life of the product because macromolecules are broken down into simpler ones, which is indicated by loss of sensory attributes in the product. Okay, then The growth of these microorganisms in the milk or milk product is governed by the environment. The microenvironment of the milk, the environment in which the milk products are stored, they will determine the microbiology of the milk and milk products. Why? Because each group of microorganisms is having its own growth requirement. If that is satisfied in the milk or milk product, we anticipate a growth of such a microorganism. Invariably, the microorganisms that are associated with the spoilage of the milk and milk product or causation of the disease in the humans, their temperature range is somewhere between 16 to 35. Okay, in this temperature window, broad spectrum of bacteria they will grow, large numbers of yeasts and mold species they will grow, leading to either spoilage of the product or causation of disease in the humans. Death may or may not happen, but the other health conditions, acute or chronic conditions, are definitely seen. We cover that under the foodborne diseases associated with foods of animal origin. There is a dedicated topic on that. Next, when it is the different sources in the supply chain of the milk and milk product that have contributed these microorganisms, they arise from animal itself, cow, air, where the animal is milked, where the milk is handled, that air quality, feeds because of the contamination, equipment that is used to handle the milk, milker or milking machine. These are all the different sources that will introduce the microorganisms into the milk, and later on they appear in the milk product also. When it comes to the total number and total kind of microorganism that we expect in the milk or milk product, it is the sum of all these. The number of microorganisms introduced by cow itself through the udder, number of microorganisms introduced through the air in the milking environment or milk handling environment, the kind of the microorganisms that are present in the animal feed, fecal or urine contamination that occurs or contamination that occurs from the body of the animal while milking process, the equipment in which the milk and milk product is handled. So all those some okay, addition, when you add all these contaminations together, the final, that is the contingent microorganism that we get in the milk or milk product is a reflection of all these contaminations occurring across the milk supply chain. The count of these microorganisms increases rapidly when there is a favorable environment. The favorable environment in the sense, most importantly, pH is no, you know that it is milk is slightly acidic, it's favorable, near to 7. Nutrients are already there. So what is required is only one factor, that is the temperature. If you can manage the temperature, to a great extent you can reduce the multiplication of the organisms. But by this temperature control, you cannot make the existing organisms zero. Clear? Even our pasteurization won't kill the saprophytic organisms. Pasteurization has been, the time temperature combination has been designed in such a way that kills the toughest of most thermotolerant microorganism in the milk, but not these contaminating organisms. When it comes to pathogens and non-pathogens, okay, the non-pathogens are having still more heat tolerance compared to the pathogens. They still survive. So even in the pasteurized milk, you will get the microorganisms that can be cultivated, but we ignore it because they are not associated with the disease. Then what change they can bring about? These organisms can bring about undesirable changes wherein they attack these macromolecules, the proteins, lipids, and carbohydrates, leading to the simpler ones, loss of sensory attributes, Final stage is end result is spoilage. So the contamination of the milk and milk products with these microflora, you have to view separately as a spoilage entity or a disease causing entity. Okay. Now the one hypothetical question that arises is, okay sir, contamination is occurring and it's very difficult to educate all the farmers, keep 100% asepsis in milking and use the sterilized equipment and all. These are very difficult and costly tasks. Can't we do another way? Okay, the other side of the coin. We'll produce the milk under unhygienic conditions because we want to attain the hygiene, but it is not attainable. We will just subject the milk to some treatment wherein all these microorganisms are killed. Is it acceptable? It is not acceptable. Why it is not acceptable? Today we are talking about the total quality management, quality assurance of the products. It is the proactive approach that should be applied from farm to fork. Okay, we are not bothered for the end product. We are bothered for the how the end product is produced. So I mean, what I look for is the achieving of the standards at each successive stage, not at the end stage alone. If it would have been permitted, probably there would not have been any clean milk production practice at all. Okay, then why is that so? While what I talked about, removal of the microorganisms from the milk is not practicable because you may remove the microorganism, okay, by centrifugation, or you may kill the microorganism by heat treatment. Okay, you can do it, but the enzymes, the metabolites produced by these microorganisms will be still stable and will bring about the pharmacological action in the pathological pathogenesis within the human consumers. That is why we should not encourage this one, and it is legally not permitted also. Okay. Rather, what we can do, we can use the clean milk production practices wherein these contaminants, microbial contaminants, will not enter the supply chain. I will use for milking purpose such a cow which is healthy and is not suffering from any infection. Utensils are clean. We educate the milk producers with respect to clean milk production practices, and will keep the product as minimal as possible. In one of the classes I have talked about, it is not possible to produce aseptic milk. Okay, milk without microorganisms is not possible because the certain lactic acid bacteria already they are there in the udder, so they are secreted in the milk. That is unavoidable. That's why it is not sterile. Milk is not sterile when you collect and when we talk about the market milk. Okay. Now we talked about various kinds of microorganisms would enter the milk supply chain. If we can understand them, what kind of microorganisms they are, obviously we can predict what kind of changes, undesirable changes they will bring about in the milk or the milk product. With that intention, these microorganisms have been categorized as microphiles, mesophiles, thermophiles, and thermoduric organisms. Okay. So what is the basis for this categorization? It is the temperature requirement of these organisms. And you be sure in your mind that these are not exclusive groups. Okay, these are groups which are having overlapping temperatures. Okay, psychrophiles will overlap with mesophiles, mesophiles will overlap with the thermophiles. However, they do have their own growth optima. So optimal temperature for the growth, it is specific for each group. Okay, the cold loving organisms, we call them as psychophiles. The room temperature growing organisms, we call them as mesophiles. heat loving organisms and heat resistant organisms, we call them as thermophiles or thermodurics. Come to the psychrophiles of the milk and milk products. These psychrophiles are cold loving organisms. Their optimal growth is 2 degrees centigrade, but in the laboratory we grow them under refrigeration temperature 7. Both gram-positive and gram-negative organisms have been incarnated in this group, and they will enter at various sources in the milk supply chain, starting from the air, utensils, the teat in the sense cow, the water. These are all the sources. These are the different gram-positive and gram-negative kinds of psychrophiles. Okay. The gram-negative organisms, most importantly, it is Pseudomonads, Acinetobacter, Flavobacterium, Achromobacter, Alkaligens, and Enterobacter. These are the major ones. Okay, this list is incomplete. We call them as Pseudomonad type of psychrophiles. Okay, if you talk about Pseudomonas is one genus in this group. That is almost 50% of all psychrophiles is made up of the Pseudomonads. Okay, they produce a heat stable enzyme. Okay, these enzymes have been identified as proteases and lipases. As the name protease indicates, it attacks the proteins. Okay, brings down from a polypeptide to the amines and ammonia, so which is a deleterious change. Proteases attacking the protein, okay, polypeptide chain. Lipases act on the lipids, milk lipids. You know that milk is rich in lipids, so brings about the rancidity and other lipolytic changes in the milk, which are undesirable. Pasteurization kills these organisms because Pseudomonas is a pathogen. Okay, given the chance, it can cause the disease in the human. So it kills the organism, but the proteases and lipases produced by Pseudomonas, they are not inactivated by pasteurization. That's why remember what I talked in the previous slides. Okay, you cannot make the milk free from the hazards when it is contaminated with the microorganisms, because you may kill the organism, but their toxins will be still active in the milk or milk product, which is having public health impact. That is why you cannot make the milk sterile at the end. Now, whenever there is a protein or fat degradation, the end products are the simpler ones such as amines, ammonia, and free fatty acids and all. They are volatile substances. You call such a product as a spoiled product because the product loses the sensory attributes, sensory quality. Okay, this is the story of the gram-negative organisms. Come to the gram-positive organisms. Various kinds of gram-positive organisms, they could be introduced into the milk supply, which are psychrophile by nature. The major group of this particular microorganism is the Bacillus species. Bacillus coagulans, Bacillus circulans, Bacillus subtilis, Bacillus megaterium. These are the major. Okay. In addition to the vegetative organisms, there are certain spores that are elaborated by these gram-positive organisms. And just like gram-negative organisms, there could be enzymes also that are produced by these gram-positive organisms. And these enzymes and spores both are thermoresistant. The pasteurization also, if it is defective, so they will not inactivate these spores. So it is an issue. Okay. And within the lab, we can cultivate these organisms between 5 to 7 degrees centigrade. And since psychrophiles are slow growers, they will take about 7 to 10 days to grow in the laboratory medium. The incubation temperature is 5 to 7. Okay. but the growth optimized 2 degrees centigrade for these organisms. But they will grow well in the lab between 5 to 7 degrees centigrade, or simply we call it as refrigeration temperature. Okay. Compared to the psychrophiles, there are many more mesophiles in the milk and milk products. Okay, milk. The normal temperature range in which these mesophiles will grow is between 20 to 40 degrees centigrade with optimal temperature of 20 degrees. Okay, within the lab we cultivate at the body temperature, that is 37 degrees centigrade, but they can be cultivated between 30 to 38 degrees centigrade within the lab. The sources of these mesophiles are many. Why many? Because the normal ambient temperature is in this range, 20 to 40. That is why many more organisms that are present in the air, present in the water, present within the cow, body temperature is also 37. Room temperature is 25. So all those pathogens, they are there in this group. They are all mesophiles, except few, majority are mesophiles. Okay. The sources, that's why they are multiple sources, and their range is also very long, very big list of microorganisms in this. Okay, to name which are very significant, this is an indicative list. Okay, some of the important microorganisms, that to bacteria, have been mentioned in this list. This list is incomplete. Micrococci, staphylococci, streptococci, microbacterium, corynebacterium, lactobacillus, bacillus, coliforms, pseudomonas. Now you see pseudomonas here. In psychrophiles also, pseudomonas is there in the pseudomonad group, and it is there here also. Okay, how come it is possible? It is possible because when we talk about the pseudomonas is a genus. There are several species in the pseudomonas. Some are cold adapted, some are room temperature adapted. So that's why it is possible. The next, third group of microorganisms that could be there in the milk and milk products are thermodurics. Okay, the temperature range of thermodurics, there is some mistake here. Okay, so there are many sources by which these microorganisms will enter again. Okay, at various points. So what are these thermodurics? So before we go to the thermodurics, let me discuss thermophiles. Okay, and I'll come to thermodurics next. The thermophiles are heat-loving organisms. Okay, they grow at higher temperatures. Their optimal growth occurs at 55 degrees centigrade. That's why we grow them on the plate count agar at 55 degrees centigrade. They can be grown up to 70 degrees centigrade. In the nature, thermophiles are present at various locations on the planet Earth. The hot springs, near volcanic eruptions. So these are all within the such aquatic ecosystems. These thermophiles are usually seen. Such a kind of thermophiles, they will be at times getting entered into the milk supply. Then you may ask a basic common question, sir, cow body temperature is 37, ambient temperature is on an average 20 to 25. From where these thermodurics will come? Okay, the certain strains of these thermophiles, they will come from the environment as an environmental contaminants. Okay, even though they have very high temperature for growth, some of the members of thermophiles can even grow at mesophilic temperature, and they will act as contaminants in the milk supply chain. Majority of the thermophiles, okay, they are all gram-positive rods. Okay, they are all gram-positive bacilli. Some of the important examples which are thermophiles are Bacillus stereothermophilus, Bacillus circulans, and Bacillus thermoacidurans. These are all some of the thermophiles that you can expect in the milk or the milk product. At times, you subject the dairy utensils to the high temperatures. The psychrophiles and mesophiles will die, but these thermophiles, since they are heat resistant, so they will still survive and they will affect the product. They may not be pathogens, but they will definitely. This group of microorganisms, that is thermodurics, these are the organisms that survive the pasteurization. Okay, they will sustain the temperature. They will sustain the pasteurization temperature and bring about the deleterious changes in the milk or the milk product. Such organisms, we call them as thermodurics. Then what is the difference between thermophile and thermoduric? Thermophiles multiply at higher temperatures, while these organisms, they will sustain the temperature and they will be surviving. Okay, they can reproduce thermophiles, they will survive. This is the difference between the thermodurics and thermophiles. So various gram-positive bacteria, bacilli, they could be the thermoduric type of organisms. Here you see, they are able to survive pasteurization, but they cannot grow. Grow in the sense they cannot multiply at that particular temperature. At various sources, these thermodurics are introduced. The microbacterium, the micrococcus, the bacillus, clostridium, fecal streptococci. These are all the examples for the thermodurics. Okay. Now, these psychrophiles, mesophiles, thermophiles, and thermodurics are 4 different kinds of the microorganisms that have been categorized based on the temperature requirement. Okay, they could enter the milk supply chain at various stages. That is why, as a practice of clean milk production, it is our duty that these organisms are not. We should see that they will not enter the supply chain. Okay. This group includes both kinds of organisms. Okay, they are either pathogens or non-pathogens. Majority are non-pathogens. More than 90% are non-pathogens. Less than say for example 10%, about 1 to 10% are pathogens. In that, this prevalence varies from region to region, from production practice to production practice, and from say for example species to species. That varies. Okay, you come to the second group of microorganisms that could be there or that could enter from the milk and milk products supply chain is the pathogens. These pathogens legally should not be there in the milk and milk products, and it is regulated by the law. Whether it is Europe, whether it is America, whether it is Australia, so everywhere this is being regulated. In India, we have got Food Safety and Standards Authority of India. Out of these pathogens, I'm going to discuss that. So some pathogens they have picked, okay, selectively, and they have mentioned in the act, and milk and milk product should be free from these organisms. Okay, these are legally important, and many times what happens, isolation and identification or detection of all these pathogens is not possible. That is why few have been mentioned in the act. It does not mean that others are permitted. Okay, it is only an indicative list that we get in the FSSAI. Okay, now let us discuss them. When it comes to bacteria, okay, large number of bacteria could be introduced by the animal itself into the milk. Example, Mycobacterium species, Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium avium paratuberculosis. They could be introduced into the milk if the animal is suffering from tuberculosis or Jones disease. Okay. Earlier, before Coxiella burnetii, this Mycobacterium tuberculosis was the index organism for or reference organism for efficiency of pasteurization. Of late, they found that even though the milk was pasteurized, there was tuberculosis which was milk-borne in some human consumers. When they investigated, okay, not tuberculosis, even though microorganism was killed, some other disease, foodborne disease was associated. Then they identified what is that, like that, and they found that this agent of Q fever, Coxiella burnetii, they found it as more heat resistant than the Mycobacterium tuberculosis. Then index organism was shifted from Mycobacterium tuberculosis to Coxiella burnetii. Okay. Now, Coxiella burnetii is the reference or index organism for efficiency, for checking efficiency of pasteurization. We have to use such a time temperature combination such that the Coxiella burnetii is completely inactivated in the milk. This is the basis. And pathogen here, the most resistant of all these pathogens is Coxiella burnetii. Okay. Now, second is Streptococcus agalactiae. When it comes to the various kinds of streptococci that are associated with contagious mastitis in animals, Streptococcus agalactiae, dysagalaceae, oberis are important. Of this agalactiae is associated with the human disease such as sore throat and all. Okay, that's why it is a pathogen. Then comes to Staphylococcus. Staphylococcus is very dangerous organism. We have coagulase positive Staphylococcus aureus, okay, and coagulase negative Staphylococci. They all could be introduced into the milk supply chain, and it is associated with staphylococcal enterotoxicosis in the humans. Even though the milk is pasteurized, the heat stable enterotoxins, okay, that are present in the milk, they are going to bring about the gastric upset, that we call it as intoxication. So Staphylococcus is a very important organism when it comes to intoxication associated with the foods of animal origin, in particular milk and milk products. You have produced the milk, okay, and you have pasteurized the milk, and you have subjected the milk for heat, okay, heat treatment, because you have prepared some product. When further processing of that product will happen, during that time, two things will happen. Enzymes, enterotoxins, they are already there in the product, or Staphylococcus aureus enters as a contaminant, so leading to food poisoning in the milk product consumers. So that's what is the speciality of the Staphylococcus aureus. Staphylococcal enterotoxin A, B, C, D, E, several kinds of toxins are there. So they are having public health problem. Okay, we'll discuss about that a little later. Then when it comes to the Escherichia coli. Okay, Escherichia coli, we have got 5 different kinds of Escherichia coli. Enteropathogenic, so enterotoxigenic, enteroinvasive, enteroaggregative, and enterohemorrhagic. So enterohemorrhagic Escherichia coli, serotype O157H7 is a cow-borne zoonosis. If the milk is not properly pasteurized, okay, or if the raw milk is consumed, that would lead to, in the human consumers, the hemolytic uremia syndrome, or hemorrhagic colitis, or TTP, thrombotic thrombocytopenic purpura. This is a condition associated with the enterohemorrhagic type of E. coli in the humans. Then Brucella abortus. Brucella, out of several species, some species are very important when we talk about the milk hygiene. Number one is Brucella melitensis. Number two is Brucella abortus. Okay, the topmost organism associated with human brucellosis is Brucella melitensis. The predominant reservoir is the goat. Then it is cattle. Okay, within the cattle, when it comes to reservoir, the cattle is reservoir for Brucella abortus. Okay, the prevalence of abortus is more, but the pathogenesis potential of Brucella melitensis is more. So these are the two organisms that could be there. Okay, how about this Brucella suis? Brucella suis and canis, it's very rare that we get them in the milk. It is mostly as a contaminant, wherein there is mixed rearing of the animals. Listeria monocytogenes. Listeria monocytogenes is a predominant psychrophile. Okay, it is a psychrophilic organism. This is a regulated organism. Our milk and milk products should be absent for Listeria monocytogenes in 25 grams of the sample, which is analyzed in the lab. And the speciality of this Listeria monocytogenes is that this is associated with an array of syndromic presentations in the human subjects. There is abortion, there is a problem with the neonates. Okay, and when it comes to the ready to eat and ready to cook type of food products, so Listeria monocytogenes is a highly regulated organism. Leptospira enterohemorrhagiae, this is one more organism associated with the animals, Bacillus cereus, Clostridium perfringens, the Yersinia enterocolytica. These are all the organisms that are pathogens. They could be introduced into the milk supply chain from the animal itself. Then when it comes to the rickettsia, the agent of Coxiella pernata is important. Okay. In addition to this, especially with respect to the goat milk, the tick-borne encephalitis virus that can cause the foodborne disease in the human consumers. That's your issue. Foot and mouth disease also that get transmitted through the milk to the humans, but it is a self-limiting disease, so it is not associated. There is no need to hospitalize the individual. That is why it goes unnoticed. Next. The human handlers can introduce large numbers of pathogens. Staphylococcus aureus, which is present in the external layers, the mucous membrane, the skin, up to the hair follicle, this organism could be there within the human handlers. All those human handlers would be introducing the organism into the milk if they won't follow the proper hygienic practices. Then E. coli, due to it is a fecal oral route of transmission through the milk or milk product here. Next is the salmonella. When it comes to salmonella, there are typhoidal salmonella and non-typhoidal salmonella. Okay, typhoidal salmonella are associated with typhoid or paratyphoid in the humans. See, non-typhoidal salmonella, say for example, Salmonella typhimurium, Enteritidis, the Dublin, the Newport, Hagana. So many 2,600 serotypes are there. Okay, they are all associated with the foodborne disease in the human consumers. So that's why Salmonella is a regulated organism. Salmonella should be absent depending on the regulation, either 25 grams or 50 grams of the food product. Shigella dysentery is associated with the diarrhea in the humans. If proper personal hygiene is not followed, the infected individual can introduce Shigella into the milk, leading to the transmission through the milk supply chain. Vibrio. Vibrio is a human disease transmitted through water. It could be introduced into the milk by the milker. Hepatitis viruses. When it comes to the largest infectious agent that is killing the human population, if at all, you see last 1 decade trends. According to WHO, it is the hepatitis virus. Okay, hepatitis A, B, non-A, non-B, like this various types are seen. Okay, they are the largest killers of the human being. In due course of time, this hepatitis virus would also cause the hepatocellular carcinoma. Okay, because one of the reason for the carcinoma development in the humans is this hepatitis virus. And if the human being won't follow the proper precautions, these viruses can enter the milk supply chain, leading to the outbreak in the human populations. However, because of the poor surveillance, what is the extent of these viral infections associated with the milk? We do not have data, especially from our Indian subcontinent. Then, fecal contamination. So, you know that there is very near proximity between the udder and the rectum or the urinary system. So there is fecal contamination that is anticipated if hygienic measures are not properly followed. And whenever there is fecal contamination that occurs, this fecal contamination, I mean, it is the mixture of both animal and the human. Okay, so how it is going to happen if the animal is tuberculosis infected? Okay, so if milk gets contaminated through the animal itself, so that becomes the source is animal. Suppose the neighboring animal is infected in a barn, neighboring animal is infected with salmonella or some other infection. If that enters this animal's milk, that we call it as a fecal contamination. Okay, so whether it is the same animal or it is a pen mate, okay, whether it is the same milker or indirectly through the other milker or something like that. So these are all the pathways by which these pathogens enter into the milk supply chain. If they enter into the milk supply chain, it is associated with the public health. They are all public health hazards. There is a public health risk. Okay. Now, some more discussion about the biology of these microflora, which is existent in the milk and milk products. Most of the microorganisms, they are very slow reproducers in the colder environment. That's why we chill the milk. We do not want them to come to the geometric ratio of milk, this reproduction in the milk. That is why the basic requirement soon after milking is chilling of the milk. Okay. The benefits of chilling the milk include the growth of these organisms gets retarded. You know that. The mesophiles, thermodurics have got higher growth optimal conditions when it comes to temperature. Okay. Except psychrophiles, rest all organisms are halted. Their multiplication is retarded. That's why we chill the milk. Okay. The chemical retardation also, the chemical deterioration also is retarded because majority of the metabolites that are produced by these organisms, they are produced invariably in the initial or mid or later stage of the log phase. Okay, log phase. That's why since the animal, this milk has not come to the such a temperature, which is the growth optima of that particular microorganism. So the production of these metabolites is inhibited. Enzyme activity is also retarded. You know that enzymes also require certain optimal conditions for the activity. So enzymatic reactions are retarded, chemical deterioration is retarded, and growth of microorganisms, okay, development and multiplication of the microorganisms is halted. Halted in the sense retarded. That is why we undertake the chilling or cooling of the milk soon after collection until it is pasteurized. Effect of cooling on the milk. Okay, microbiology. The freshly drawn milk is warm to touch, 38 degrees centigrade. Okay, it is 37 or 38 depending on the condition. Okay, these organisms at that particular temperature will rapidly grow. When you store the milk, okay, with the air temperature of around 16 degrees centigrade, okay, up to 3 hours, milk stored in a shade, okay, the air temperature is around 16 degrees. It will take around 3 hours to reach to that temperature. Then this scenario what I have depicted here may not be applicable always. Okay, so 3 hours we should not keep the milk at all. Thereon, even if you keep the milk, the deteriorative changes will increase in their quantum, affecting the shelf life of the product. Okay, there is a built up of the acidity that would lead to the coagulation of the milk, or that will induce such milk undesirable. Okay. So, if you keep the milk at around 16 degrees centigrade, the time required, okay, to fall from 38 degrees to 16 degrees is 1 hour. Okay, when you cool the milk with cold running water. So these are the scenarios I am presenting here. Okay, these may not be applicable under actual field conditions. So when the milk temperature is dropped from 38 to 16 degrees centigrade, there is a certain reduction in the growth and development of the microorganisms. So this is one more depiction what happens if the temperature further goes down, what we have to do. That's the thing. Okay. As we understand by our discussion, the milk contamination can occur at the various stages in its production, processing, and distribution. The first thing we need to do is we have to keep the animals healthy, so that whatever the microbial load they set in the milk, that gets reduced. We have to keep them disease free. Okay, we have to milk the animal under hygienic conditions, and we have to maintain the equipment hygiene properly. Okay, so that they won't contribute to the total contingent number of these microorganisms in the milk. Okay, then for that, what can we do? There are two things that need to be done. What is that product is, and accordingly what kind of analysis we have to do in the laboratory to know the count. Okay, so again in the practical I'm going to discuss about this in detail, but at this point of time, if it is a raw milk, we have to test its general quality. Okay, general quality starts with organoleptic evaluation, counting these microorganisms using direct microscopic count. How many individual bacteria or yeast or mold are there, and how many clumps of such organisms are seen. Somatic cell count in the milk. This somatic cell count is nothing but lymphocytes, neutrophils, macrophages, and eroded or excoriated epithelial cells that come from the teeth. Okay, there is a threshold number. Invariably, we say that around 2 lakh per ml is permitted. When the count increases beyond 2 lakh, it indicates some udder infection. Okay, mastitis. So that's why somatic cells indirectly will indicate whether the udder is healthy or non-healthy. Then for shelf life, we have to undertake a preliminary incubation test. Just you take the product and keep at room temperature. You see how long it is going to sustain. That's what it is. Or specifically, if you want to undertake the microbial count, as we talked in the beginning, these microorganisms are either Microphiles, Mesophiles, Thermophiles, or thermodurics. We have to undertake the count of all these organisms using suitable media, following appropriate time temperature combinations for the incubation. Okay. If it is a pasteurized milk, one thing we can undertake the shelf life assessment by just like preliminary incubation, what we talked about, or other suitable laboratory test, or we can count the organisms, okay, in the milk. Then dried milk products. Dried milk products, microbial count can be performed. All these, what we talked earlier, in addition to that, yeast and mold can also be counted. Butter. Butter, we can undertake the microbial count. In addition to the routine test, we can go for lipolytic organisms count, proteolytic organisms count, yeast and mold also. Frozen dairy products, we can undertake the microbial count in terms of bacterial count as standard plate count and yeast and mold count. Concentrated milk products, we can go for microbial count as above. The fermented products, okay, including cheese, yogurt, and other fermented products, we can undertake the microbial count. When it comes to the microorganisms and the milk and milk products, in India, the Bureau of Indian Standards has given certain specifications. Okay, these specifications are like this: pasteurized milk, standard plate count 30,000 maximum. Coliforms, they should be absent. Condensed type of milk products, SPC maximum 500 gram, and coliform should be negative. Yeast and mold 10 per gram. Milk powder maximum count, SPC 5000 per gram, coliform 90 per gram. Ice cream 2.5 lakhs, SPC, and coliform 90 per gram. These are the specifications given by the Bureau of Indian Standards. In Europe, European standards are applicable. Different directives, council directives, and regulations, they are applicable for that. Okay. So it all depends on the what kind of country it is. So what kind of these SPS measures they expect. So that's the thing that decides the critical limits for these microorganisms. Okay. So Codex at this point of time is only looking after the chemical residues. In due course of time, it will definitely specify microbial limits also for various kinds of foods of animal origin. However, we have ICMSF, which is a non-governmental, not-for-profit organization that has given certain specifications for foods of animal origin, including milk, and we have milk and milk products, and we have different sampling plans under this ICMSF, 2-class plan and 3-class plan. Regarding this, we'll discuss in subsequent classes. That's all about the microbial flora of milk and milk products, and somewhere I highlighted some of the important foodborne pathogens that are associated with foodborne disease. In the subsequent class, what we will do, we will discuss the individual pathogen and the effect or impact caused by these individual organisms. Okay, in what way they are going to affect like that.